Long before dinosaurs shook the earth, evolution tried out creatures so strange, so experimental, and so unlike anything alive today that they seem pulled from another universe. Fractal quilts drifting on ancient seafloors, boomerang-headed swimmers, spiny walking cacti, armoured river titans, and saber-toothed proto-mammals ruling a world on the brink of collapse. These weren't the warm-ups for dinosaurs, they were entire ecosystems of evolutionary prototypes, each rising to dominance before disappearing forever. This is the story of the hidden evolutionary experiments that failed before dinosaurs, and how their vanished designs shaped the world that came after them. Rangeomorphs, Ediacaran, quilts. Rangeomorphs are some of the weirdest animals that have ever lived on Earth. They were around long before anything with mouth, eyes, or legs came into being. It was between 575 and 541 million years ago, during the Ediacaran period. This is tens of millions of years before the Cambrian boom. They look like plants or frilly sea flowers at first glance. However, they were neither plants nor animals as we know them. They were a failed experiment in evolution. They were a branch of life that grew into huge bodies with fractal patterns, ruled whole environments for millions of years, and then went extinct without leaving any offspring. Rangeomorphs didn't move, they didn't hunt, they didn't even eat the way animals do today. Instead, they absorbed nutrients directly from seawater. Their bodies were made of repeated branching fronds, each one shaped like a feather made of smaller feathers, which were made of even smaller feathers. This fractal design, structures repeating at different scales, is something no modern animal uses. It allowed rangeomorphs to maximize surface area, which helped them take in dissolved nutrients in oceans that were rich in organic material but poor in oxygen. Fractophosus is one of the most famous rangeomorphs. It was found in the famous Mistaken Point fossil beds in Newfoundland. Whole fields of these organisms have been kept in place, showing how they spread out across the seabed like huge mats of living things. By Ediacaran measures, some people grew to be more than a metre long, which makes them giants. But the fossils also show something very interesting. They spread in two ways at the same time. They put out tiny eggs that floated away, and clones that grew around the parent were also made. They were good at colonising because they used this double strategy. They were one of the first examples of a complex breeding system in Earth's past. Other rangeomorphs, like Charnia, stood tall like underwater ferns anchored to the sea bottom. They didn't photosynthesize despite looking plant-like. Instead, they lived in deep, dark waters where sunlight couldn't reach. Their food came entirely from dissolved organic compounds drifting through ancient seas. This feeding method is so unusual that scientists still debate what group rangeomorphs belong to. Some think they were early animals unlike anything living today. Others argue they were something in between animals and fungi, a now extinct experiment that explored a totally different way of living. What makes rangeomorphs especially interesting is that they represent a type of body plan evolution would never revisit. After the Ediacaran ended, nothing with their fractal structure or carpet-like ecosystems ever returned. When the Cambrian explosion kicked off, animals evolved muscles, mouths, guts, and hard parts. Predators appeared, mobility became important, and rangeomorphs, soft, static organisms that depended on nutrient-rich water, couldn't keep up. Their world vanished. One big reason for their disappearance may have been the change in ocean chemistry. As oxygen levels rose globally, nutrients became more limited in shallow waters. Rangeomorphs had thrived in low oxygen, high organic conditions. When the environment shifted, their feeding style stopped working. Meanwhile, early animals that could move, graze, and hunt started taking over the seafloors. Another possible reason is predation. During the late Ediacaran, fossils show signs of early grazers, small organisms that scraped surfaces for food. These new animals may have damaged or consumed rangeomorph tissues. Even light grazing pressure could be devastating to creatures that couldn't move or heal effectively. The arrival of predators in the Cambrian almost certainly accelerated their extinction. Rangeomorphs helped shape Earth's early ecosystems, even though they lived in strange ways and eventually went extinct. They made sediments more stable, added air to the area, and set up the first large-scale groups of complex life. A lot of years before animals came along, they were kind of like Earth's prototype for big complex creatures. They tested what was possible. Dickinsonia and the pancake fauna. Dickinsonia is one of the most famous secrets of the Ediacaran period. It is an oval, flat, ribbed organism that looks like a living bath mat pressed into mud from the bottom. Between 558 and 550 million years ago, it lived. That's a long time ago, before animals with bodies, shells, or even heads had formed. Its soft body left marks that are so clear that you can count each rib. But scientists still can't figure out where it fits in the tree of life, even though they have thousands of fossils. Many paleontologists refer to Dickinsonia as part of the pancake fauna. This is a group of flat, quilted organisms that don't fit into any current categories and are thought to be evolutionary tests that ended before the Cambrian explosion. At first glance, Dickinsonia seems simple. It had no mouth, no gut, no eyes, and no limbs. Its body was divided into repeating segments that curved outward like the steps of a nautilus shell, though they weren't true segments in the animal sense. These modules give Dickinsonia a bilaterally symmetrical shape, but not the kind of symmetry we see in modern worms or insects. Instead, its symmetry is offset, almost like a zipper laid slightly crooked across its centre. This strange design hints that Dickinsonia wasn't just unusual. It may have belonged to an entirely extinct branch of multicellular life. The way Dickinsonia is fed is what makes it captivating. Feeding traces, shallow depressions left behind when the animal sat on microbial mats, can be found in fossil beds in Australia and Russia. The bottom was covered in these thick mats of bacteria and algae. It looks like Dickinsonia ate these mats from the outside, kind of like how you could lie down on a pizza and take in the toppings through the crust. It left behind cleaner areas where the layer of microbes had been removed as it moved. This is not how animals eat today, but it makes sense in a world where bacteria mats cover the seas. One of the biggest scientific breakthroughs came in 2018 when researchers found traces of cholesterol, a diagnostic biomarker for animals, in Dickinsonia fossils from the White Sea region. This discovery strongly suggested that Dickinsonia was an early animal rather than a fungus, lichen, or something in between. But even with this chemical clue, Dickinsonia still doesn't fit into any modern animal group. It lacked a mouth, intestines, or organs typical of even the simplest worms. Some scientists propose that it belonged to a now extinct grade of early animals that branched off before modern animal groups formed. Dickinsonia didn't live alone. Its world included other flat-ribbed organisms like Yorgia, Andiva, and Kimberella, the pancake fauna. These organisms covered the shallow seafloor like soft, living tiles. Some stayed anchored in place, others moved slowly, leaving faint tracks. Together, they formed the first complex ecosystems. Yet none of these organisms survived past the Ediacaran. Their unique bodies and lifestyles made sense in their environment: soft seafloors, rich microbial mats, and stable conditions. But once the Cambrian explosion introduced burrowers, grazers, and predators, the pancake fauna collapsed. Changes in sediment structure also played a major role in their downfall. During the Ediacaran, microbial mats held seafloor sediments together like glue. When early Cambrian animals evolved ways to burrow and churn through mud, these mats began disappearing. Without them, Dickinsonia's feeding style no longer worked. The soft seafloor also became more disturbed and unpredictable, making it harder for delicate, flat organisms to stay intact. Evolution was reshaping the ocean floor into a place that favoured mobility, skeletons, and active feeding, things Dickinsonia never evolved. It's also unclear how Dickinsonia multiplied. Some skeletons show people of different sizes squished together, which could mean that they grew quickly or had babies. Some have designs that look like they are budding, which suggests that new creatures may have grown from older ones. But no fossil shows clear signs of reproductive systems. Dickinsonia lived in a time before fossils of eggs, shells, or embryos were found, so one of its mysteries is still how reproduction works. Helicoplacoids, spiral urchins. Helicoplacoids are some of the strangest and shortest-lived evolutionary experiments ever found in fossils. They were sea animals with bodies that looked like spinning tops and were covered in rotating plates. There is no other animal like them living today. They suddenly showed up in the early Cambrian, about 520 million years ago, did well for a short time, and then went away for good. Scientists had a hard time putting them in a larger group for decades. In the end, proof showed that they were early echinoderms, from the same family tree as starfish, sea urchins, and sea cucumbers. However, they broke almost every rule for echinoderms while they were doing this. Unlike modern echinoderms with 5-part symmetry, helicoplacoids had a helical body structure. Their plates spiraled around the body in a twisting pattern that looked almost like a seashell made of interlocking armour. This spiral structure wasn't just for show. It allowed the plates to expand and contract as the animal moved or fed, something no living echinoderm can do. The plates were arranged so flexibly that the body could subtly twist or bulge, almost like an accordion made of stone. This unique design suggests helicoplacoids may have been adapted to shifting currents or soft seafloors, where stability was more important than speed or complexity. The creature's mouth sat near the top, and the spiral plating likely helped channel water and food toward it. Helicoplacoids probably fed on suspended particles or organic debris drifting through the early Cambrian seas. Some scientists believe they may have rotated slightly while feeding, using their spiral grooves to direct nutrients toward the mouth. Others argue they simply lay on the seafloor, letting water currents bring food to them. Either way, they lack the complex feeding arms or jaw structures seen in later echinoderms. One of the biggest clues to their lifestyle comes from where their fossils are found. Helicoplacoids appear almost exclusively in rocks formed from rapidly shifting, storm-disturbed environments, especially in what is now Western North America. These habitats were unstable, with shifting sands and frequent sediment slumps. Helicoplacoids may have been specialized for living in this kind of dynamic setting, where their flexible, spiraling bodies could help them anchor or adjust to changing currents. But that specialization came with a cost. As the Cambrian seas evolved, environment stabilized, burrowing animals became more common, and seafloors grew more disturbed by new forms of life. These changes may have wiped out the soft, unstable habitats helicoplacoids depended on. When the seafloor began to churn under the activity of early worms and arthropods, the delicate balance that allowed helicoplacoids to thrive collapsed. Their extinction may have been less about competition and more about losing the specific conditions they needed. Even identifying helicoplacoids took decades of scientific debate. Their odd symmetry baffled paleontologists because it didn't match any known animal pattern. Echinoderms are famous for their 5-part symmetry, yet helicoplacoids completely ignored this rule. Only when researchers examined how their plates grew and compared them with early echinoderm fossils did the connection become clear. The arrangement of calcite plates and the microstructure of their skeleton revealed that helicoplacoids were indeed early members of the echinoderm family tree, just an experimental branch that tried a radically different approach to body design. Their end shows an important idea from the early stages of evolution. Not all good designs last. Helicopteroid plants only live for a few million years, which is a very short time in natural terms before dying out. Other echinoderms changed their bodies to be more stable and adaptable so they could survive major extinctions, competition, and changes in their surroundings. Helicoplacoids couldn't survive in the fast-changing Cambrian world because they had a small biological niche and an odd structure. Stylophorans, carpoid echinoderms. In the echinoderm family, stylophoran were some of the most unpredictable animals. They lived in the early Paleozoic and disappeared long before dinosaurs did. There is a rule that most echinoderms follow, and if you know anything about starfish, sea urchins or crinoids, you know it. It didn't matter at all to stylophorans. Their bodies were skewed, covered in metal plates, and built around a single, long limb that experts are still not sure what it did. No one could agree on what they were for decades because they didn't look like their cousins. Some people thought they were missing connections between echinoderms and chordates. Others thought they were failed attempts with echinoderms. Most paleontologists now agree that they were echinoderms, but because they broke so many natural rules, they could never have lived for a long time. A typical stylophoran had two main parts. The first was a flattened, asymmetrical body, called a theca, covered in interlocking calcite plates that acted like armour. This body didn't show the usual radial layout seen in starfish or urchins. Instead, it looked more like a small shield with no obvious front or back. Attached to this theca was a long appendage called the olacophore, built from a series of calcite elements arranged like a flexible rod. This structure is the heart of the stylophoran mystery. Was it a tail, a feeding arm, a respiratory organ, or something totally unique? For decades, scientists argued fiercely about the olacophore. Some early researchers believed it was a proto-spine, making stylophorans early relatives of vertebrates. This idea came from the fact that the appendage had segments that superficially resembled a notochord. Others countered that the olacophore worked more like a feeding arm used to sweep particles toward the mouth, similar to structures in some living echinoderms. Modern imaging techniques finally tipped the scales. The microstructure of the plates matched echinoderm patterns, not vertebrate ones. It wasn't a backbone precursor, it was echinoderm tissue redesigned into a unique tool. We now know the answer to that question, but stylophorans are still unusual. Just the fact that they aren't flat makes them stand out. Echinoderms are known for landing on 5-part symmetry early in their development, but stylophorans didn't follow that plan at all. Why? One idea is that stylophorans emerged before the group's 5-part symmetry became set. This means that they split off early and kept a body plan that was more flexible and old. Asymmetry might have helped them scrape the bottom or stick to soft sediment, which is another option. Stylophorans also raise questions about how they live. Some species show signs of being deposit feeders, sweeping food toward a mouth located on the theca. Others may have stood partly upright, using their olacophore to capture suspended particles in the water. A few fossils seem to show individuals attached to hard surfaces, suggesting they might have been able to anchor themselves temporarily. The enormous variety within the group hints that stylophorans were trying many different strategies, none of which lasted beyond the Ordovician. What ultimately doomed them? Part of the answer lies in the rapidly evolving ecosystems of the early Paleozoic. During the Ordovician, the oceans filled with new predators, including early cephalopods and jawless fish. These hunters could crush or pierce armor plates that had protected earlier animals. Stylophorans, with their awkward shapes and limited mobility, may have been easy targets. At the same time, other echinoderms shifted toward more efficient designs: starfish with flexible arms, sea lilies with filter-feeding crowns, and urchins with protective spines. Stylophorans simply couldn't compete. Changes in ocean chemistry may have added further pressure. As reefs expanded and seafloors became more oxygenated, organisms that relied on slow feeding methods or stationary lifestyles faced increasing disadvantages. Stylophorans were likely too specialized to adapt. Their asymmetry, once an experiment in form and function, became a liability. Radiodonts. Radiodonts were the first real giants and top predators in the Cambrian seas. They were strange, alien-looking animals with facial claws that could grasp things, mouths that looked like circular saws, and bodies that were made for fast, efficient swimming. They showed up more than 520 million years ago, ruled the world's seas, and then disappeared long before dinosaurs did. The most famous one, Anomalocaris, was up to a metre long, which was very big for its time. It changed everything we knew about how early animals looked. Radiodonts were hunters, but they did other things too. They were a whole group of genetic tests that looked at body plans, ways of eating, and sensory systems. They would go extinct without leaving any straight heirs. The defining feature of radiodonts was their frontal appendages, long segmented arms located in front of the mouth. In Anomalocaris, these arms curved inward and ended in spines used to snatch prey. Earlier reconstructions once mistakenly placed these appendages on different animals, which is why Anomalocaris literally means strange shrimp. Only later did scientists realize the shrimp was just one part of a much larger creature. CT scans of Radiodont fossils show that these appendages weren't just claws. They were multi-purpose tools, capable of grabbing, sweeping, and even probing the seafloor depending on the species. The mouth of Radiodonts was another bizarre innovation. Instead of jaws, it had a circular ring of plates that opened and closed like a rotating pineapple slicer. For years, paleontologists believed this mouth could crush trilobites, but new studies show it may not have been powerful enough to crack hard shells. Instead, many radiodonts likely targeted softer animals or scavenged debris. Some species had more flexible mouth plates, suggesting they filtered food rather than sliced it. This variety shows radiodonts were not a single lifestyle. They were dozens of evolutionary experiments under one umbrella. Not all radiodonts were hunters, which was one of the most important findings in the last few years. Some of them, like Tamisiocaris, had very long filtering parts that looked like whales' baleen plates. These animals moved with their mouths open, pulling small things out of the water. This means that radiodonts figured out how to filter feed hundreds of millions of years before whales did, but their bodies were built very differently. Not only were there hunters during the Cambrian, but there were also early giants that ate plankton. Radiodont bodies were built for movement. Their sides were lined with overlapping swimming flaps, an early version of the undulating fins seen in cuttlefish today. These flaps created a smooth, wave-like motion that pushed them forward with surprising speed and control. Fossil beds show radiodonts often lived in areas with strong currents, where their swimming ability allowed them to travel long distances and hunt efficiently. Their eyes were advanced for the time, with some species showing up to 30,000 lenses, indicating high-resolution vision. In an ocean filled with slow-moving and poorly defended animals, radiodonts were sensory powerhouses. So why did these apex predators vanish? The answer lies in both environmental change and competition. As the Cambrian gave way to the Ordovician, ecosystems became more structured and diverse. Trilobites grew tougher. Early jawless fish evolved better armor and faster swimming. New predators, such as cephalopods, entered the scene with shells and jet propulsion. Radiodonts, with their soft bodies and exposed frontal limbs, suddenly found themselves outmatched. Their once dominant position in the food chain eroded as prey became harder to catch, and competitors became stronger. Another problem was ecological specialization. Filter-feeding radiodonts needed stable plankton levels, and shifting ocean chemistry may have disrupted their food supply. Predatory species relied on open water ecosystems that were changing as reefs expanded and seafloors grew more complex. Radiodonts were built for a specific world, a world that gradually disappeared beneath them. Still, radiodonts left a mark even after they died out. Their unique body design, which included frontal limbs, complex eyes, and bendable fins, made them the first major predators in animal history. They were the first people to hunt with their eyes, swim long distances, and eat specific foods. They didn't have any straight offspring, but they did play a part in the evolutionary arms race that pushed animals to make shells that were harder, swim faster, and have better senses. Hallucigenia and armored lobopodians. One of the most well-known images of early evolutionary research is the hallucigenia, a creature that was so strange that scientists spent decades putting it back together backwards and upside down. It was a lobopodon, which is a larger group of worm-like animals with soft bodies and short legs that lived on the Cambrian seabed more than 508 million years ago. These animals were related to velvet worms in the past, but their body plans were much stranger and more varied than those of living things today. Lobopodians made the Cambrian a playground for weird creatures, with backs covered in spines, metal plates, and eye stalks. These forms did well for a short time before going away for good. Hallucigenia itself is the best known of the group, discovered in Canada's Burgess Shale. Its body looked like a thin tube with multiple pairs of soft legs underneath and rigid spines sticking out from its back. When paleontologists first studied it in the 1970s, they placed the spines downward as legs and the soft limbs upward as tentacles. A reconstruction that made Hallucigenia look like a floating balloon animal. Only in the 1990s did scientists flip it the right way up, and in 2015, high-resolution imaging finally revealed its head, solving one of the strangest puzzles in paleontology. What makes Hallucigenia so interesting is what it tells us about the early stages of arthropod evolution. Lobopodians, like Hallucigenia, Microdictyon, and Collinsium, had a mix of features seen in modern velvet worms, early arthropods, and entirely extinct lineages. They had stubby legs without joints, simple eyes, and flexible bodies. But they also developed armor, such as rows of spines, scales, or sclerites. These features were early tests of the protective designs that would eventually lead to arthropods, creatures like trilobites, insects, and crustaceans. Take Microdictyon, another lobopodian known for its distinctive, net-like armor plates. These plates sat on the sides of its body like small shields. They were not quite exoskeleton segments, but they hinted at how early animals experimented with protection. Collinsium, sometimes called the walking cactus, took this even further with spectacular spines covering its entire body and long, delicate feeding appendages extending from its head. Each species tried different mixes of defense and feeding strategies, showing how flexible early evolution was in designing new creatures. A noteworthy discovery is that some lobopodians had claw-like structures at the ends of their legs, made of stacked proteins that look a lot like the claws of current arthropods. This means that claws and arms on arthropods probably didn't all appear at once, but rather changed over time from much simpler forms. Lobopodians were the first animals to test these new ideas. Their legs could hold on, grip, and secure, but not as well as the legs of real arthropods. Hallucigenia's spines may have protected it from early predators like radiodonts, which were becoming more efficient hunters during the Cambrian. Its narrow, tube-like body likely helped it hide among microbial mats or soft sediments. It didn't move quickly. It probably scraped food from the seafloor with simple mouthparts. This slow lifestyle made armor essential. Lobopodians that lacked protective features often disappeared sooner or evolved new traits to stay ahead of predators. But even with these adaptations, Lobopodians did not last as major players. By the late Cambrian and early Ordovician, arthropods had evolved segmented bodies, hardened exoskeletons, and jointed limbs. These improvements allowed them to move faster, protect themselves better, and adapt to many habitats. Lobopodians, with their soft bodies and limited mobility, were outcompeted. Most lineages died out, leaving only distant relatives like today's velvet worms and tardigrades. Recent fossil discoveries from China's Xinjiang biota reveal that lobopodians were far more diverse than once thought. Some had multiple, long feeding arms. Others had strange mouth cones or elaborate sensory organs. These fossils show that lobopodians explored many evolutionary ideas before settling into the small niche their modern descendants occupy. The armed cousins of Hallucigenia are a key example of how evolution has been created. They weren't fails or mistakes. They were just samples. Their work with protection, arms, and sensory systems set the stage for the powerful arthropods that would later take over Earth. But in the end, their strange line disappeared, leaving only fine-grained mudstones that showed how they looked. Waiwaxia and Halkyrids, chainmail slugs. Waiwaxia and the Halkyrids were some of the weirdest animals to appear in the early Paleozoic. This was a time when evolution was trying out a lot of different body types, armor, and tools for eating. These animals were soft-bodied slugs with spines and scales that overlapped to protect them, like live versions of armor. They showed up in the Cambrian, did well for a short time, and then disappeared, leaving no living relatives today. In spite of this, their remains show an important part of the history of evolution, the first steps towards shells, armor, and other protective structures that would later define mollusks, brachiopods, and other major animal groups. Waiwaxia, found in Canada's Burgess Shale, looked like a flattened oval slug covered in rows of leaf-shaped scales called sclerites. These sclerites overlapped like shingles on a roof, forming a flexible armor that protected the soft interior. Along its sides and back Waiwaxia carried long spines that likely discouraged predators like Anomalocaris. Its underside was soft and unarmored, showing the early stages of a creeping foot similar to what we see in modern mollusks. But Waiwaxia's mouth was the real surprise, a pair of hooked, scraping elements that resembled early versions of the molluscan radula, a tongue-like organ covered in tiny teeth. This radula-like structure has sparked some of the biggest debates in paleontology. Was Waiwaxia an early mollusk, testing out the first scraping mouth? Or was it part of a completely separate lineage that simply evolved similar feeding tools? The answer still isn't agreed upon. Some fossils show that the feeding structures in Waiwaxia were simpler than true radulae. Others show enough similarity to suggest a direct connection. Either way, Waiwaxia represents one of the earliest attempts at specialized feeding tools, something that became essential in later marine ecosystems. Halkyriids, such as Halkyria and Walcottidiscus, shared some features with Waiwaxia, but had a very different shape. Instead of being oval and flat, they had long slug-like bodies with armour arranged in two major sclerite zones, one on each side, forming overlapping chainmail plates. At their front and back were two hard shells, like small cap-shaped shields. This three-part design, shell, armour, shell, has fascinated scientists because it resembles the arrangement seen in early mollusks, particularly chitons, which also have repeating armour plates. But Halkyriids were not direct ancestors of modern chitons. They were parallel experiments, exploring similar ideas but heading down a branch that would eventually disappear. The form of their shells and sclerites is one of the most important clues about how they lived. Halkyriid sclerites had small pits and lines that showed they kept growing and were changed as the animal aged. Their shells had growth lines that were like those of early brachiopods and mollusks, but not the same. Because of this mix of traits, scientists call Halkyriids mosaic organisms, animals that had traits from several animal groups but didn't clearly belong to any of them. They were prototypes that mixed protection, soft bodies, and slow movement in ways that no living thing does today. Both Waiwaxia and the Halkyriids lived in ecosystems filled with early predators. Radiodonts, large arthropods, and even small worms were learning to bite, crush, or scrape food from the seafloor. In this dangerous world, having armour was one of the most important strategies for survival. The success of Waiwaxia and Halkyriids during the Cambrian shows how valuable protective structures were becoming. But as time went on, predators evolved stronger jaws, faster bodies, and more precise hunting tools. The flexible armour that protected these early slugs simply couldn't keep up. More advanced mollusks developed thicker shells. Arthropods created jointed exoskeletons. Halkyriids and Waiwaxia, with their soft underbellies and limited mobility, lost the evolutionary race. Their extinction was gradual but certain. By the Middle Cambrian, they were already declining. By the Ordovician, they were gone. Their designs may have been too specialized or too fragile to survive long-term environmental changes. Or they may have been outcompeted by animals that refined the ideas they only hinted at: true radulae, proper shells, and stronger protective plates. Trilobites. Many people think that trilobites, which were sea arthropods that lived for more than 270 million years, evolved into thousands of species and lived in all of Earth's oceans, is one of the best examples of evolution at work. But even with all of that success, trilobites are one of the best examples of an experiment in evolution that failed completely. They suddenly arrived at the start of the Cambrian period, learned to do almost every biological job there was, and made some of the most advanced eyes ever found in fossils. During the Permian mass extinction, they were gone forever, leaving no descendants. It's clear from their story that even the most flexible plans can fail when things change too quickly for living things to adjust. At their core, trilobites were built around a simple plan: a hard exoskeleton, a three-part body, and jointed legs hidden beneath protective plates. This exoskeleton was made of calcite, the same mineral that makes up limestone. It gave trilobites incredible preservation potential, one reason why we have so many fossils today, but it also gave them a natural defense against Cambrian predators. Their bodies were divided into 3 regions: the cephalon, head, thorax, middle, and pygidium, tail. These segments allowed trilobites to roll into a tight ball, a defense strategy still used by modern pill bugs. This ability made them early winners in a world filled with newly evolved predators like radiodonts. The eyes of trilobites were one of the most intriguing data about them. Many species developed lenses made of calcite, arranged in complex designs that gave them great vision. Some had eyes with thousands of individual lenses, allowing them to sense movement with amazing sensitivity. Others evolved tower eyes, rising from their heads like periscopes, letting them see while buried in the ocean. These arthropods' visual systems were so advanced that no other group ever made something like them. Trilobite eyes were an evolutionary high point that surprisingly left no future impact. Trilobites explored almost every lifestyle available in ancient seas. Some burrowed under sediment looking for food. Others swam in open water. Some scraped algae, while others hunted small animals. There were species with spines, horns, ridges, or smooth bodies designed for fast swimming. There were giant trilobites over 2 feet long, like Isotelus rex, and tiny ones only a few millimeters across. This diversity helped trilobites survive multiple extinction events during the Cambrian and Ordovician, but it couldn't save them forever. Their decline began long before their final extinction. During the Devonian, jawed fish became major players in the oceans. These fish were faster and stronger than anything trilobites had ever encountered. Their jaws could crush trilobite shells, and their growing dominance pushed trilobites into fewer ecological spaces. At the same time, ammonoids, ancient relatives of octopuses, rose as swift and agile predators. Trilobites that once dominated seafloors found themselves squeezed by new competition from above and below. Another challenge came from changing ocean chemistry. Trilobites relied on calcium carbonate to form their exoskeletons. During periods when oceans became more acidic or when carbonate levels dropped, trilobites struggled to form shells. Environmental shifts like these occurred repeatedly through the Paleozoic, and each event reduced trilobite diversity a little more. By the late Permian, trilobites were hanging on in only a few environments. The rise of large marine reptiles, new predatory fish, and increasingly unstable oceans pushed them into a corner. When the end Permian mass extinction struck, triggered by massive volcanic eruptions, ocean acidification, and runaway climate change, trilobites were already weakened. They disappeared completely after more than 250 million years of success. It's strange that they went extinct because other invertebrate groups lived on. Crabs, horseshoe crabs, and early insects made it through. It looks like trilobites were too specialized in how their bodies were built and too reliant on water chemistry that no longer supported their mineralized shells. As far as their genetic experiment went, it was over. Graptolites, planktic colonies. In the early Paleozoic, graptolites were some of the most common and successful animals. They were also one of evolution's strangest experiments, with colonies of very small zooids living together in structures that swung or floated and looked like saw blades, tuning forks, or fine calligraphy carved into stone. For more than 100 million years, they ruled the oceans and swam through the old seas as good filter feeders. Eventually, they went extinct, leaving only a small group of survivors hidden among modern hemichordates. Their rise and fall shows how even animals that don't look like much can change whole ecosystems and then disappear when the ocean conditions change around them. A typical graptolite wasn't a single animal, it was a colony, made of dozens to thousands of tiny individuals called zooids, each living in a small cup-like structure called a theca. These thecae connected into long branches called stipes. Early graptolites attached themselves to the seafloor, but by the Ordovician, many species evolved into free-floating plankton, drifting in the open ocean like living ribbons. This shift from benthic to pelagic living helped them spread across the world. Graptolites became so abundant and widespread that they are now some of the best index fossils geologists use to date Paleozoic rocks. The most fascinating facts about graptolites is that they were hemichordates, which means they were related to oakworms and pterobranchs living today. Pterobranchs are tiny animals that live in tubes and can still be found in the seas today. They form groups that look like small versions of graptolite structures. Scientists didn't prove this link until the 20th century, when they studied their genetic patterns and soft tissues. Before that, paleontologists didn't agree on whether graptolites were algae, plants, or something else. Their fossils were so simple, just dark lines made of carbon, that it took decades to figure out what they were. Graptolites succeeded because their body plan was perfect for Paleozoic oceans. Their colonies were lightweight, flexible, and efficient at capturing drifting food particles. Many species developed elaborate branching patterns with multiple stipes extending outward like the teeth of a comb. These patterns increased surface area without requiring more material. Some colonies grew into spirals. Others formed zigzagging ribbons. Each shape was an experiment in maximizing feeding efficiency while staying afloat in the water column. One of the biggest evolutionary advantages graptolites had was their ability to adapt quickly. Fossil records show rapid changes in colony shape, thicker size, and branching patterns over short geological periods. These shifts let them respond to changes in sea temperature, nutrient availability, and ocean circulation. Graptolites were early masters of ecological flexibility, which is why they weathered several early extinction events without disappearing. But their success also depended on a very specific set of ocean conditions. Graptolites thrived in nutrient-rich waters with strong upwellings and stable plankton communities. As long as oceans circulated in predictable patterns, graptolites spread across the world. Their decline began in the late Silurian and Devonian, when ocean chemistry and circulation patterns changed dramatically. Reefs expanded, oxygen levels shifted, and new predators evolved in the plankton. These changes filtered slowly through the food web, disrupting the delicate balance that graptolites depended on. Competition also played a role. Other planktonic animals, such as early crustaceans, improved their feeding strategies. Some developed more efficient filtering methods. Others evolved harder bodies that resisted predation better. Graptolites, with their soft tissues and simple feeding structures, could not match these new arrivals. Their colonies became less diverse, their geographic range shrank, and by the Devonian, many lineages were already fading from open water ecosystems. During the late Devonian extinction events, the last piece of the puzzle fell. The oceans became less stable, oxygen levels dropped in many places, and the rhythms of ocean movement that the oceans relied on stopped working. Most graptolite groups are no longer there at all. Only a very small group of animals, ancestors to modern pterobranchs, survived. They lived in safe places away from the rough open seas where millions of colonies used to live. Euryterids, sea scorpions. Euryterids, which are sometimes called sea scorpions, ruled rivers, lakes, bays, and even the shallow seas for more than 200 million years. They were some of the scariest arthropods that ever lived. They first showed up in the Ordovician period and got really big in the Silurian. Before sharks and bony fish took over, they were one of the most important hunters. Other species, like Jacalopterus, were more than 8 feet long, making them the biggest arthropods in Earth's past. Some species were small and quick. Their rise and fall tell a story of power, change, competition, and ultimately failure. It's the story of an evolutionary path that worked well for a long time, but couldn't handle the stresses of a world that was changing. At first glance, Euryterids look like something out of a nightmare. Elongated bodies, segmented tails, powerful pincers, and large compound eyes set on flat, shield-like heads. Their bodies were divided into a prosoma, front, and opisthosoma, rear, with a long, flexible telson at the end. In some species, the telson was a broad paddle used for swimming. In others, it was a sharp spike that may have been used for defense or for anchoring themselves while feeding. Euryterids were incredibly diverse. Some crawled along the seafloor like armored lobsters. Others swam gracefully like underwater insects, and a few were streamlined pursuit hunters capable of chasing prey with speed. One of their major evolutionary strengths was their versatile limbs. The first pair of legs often ended in powerful pincers used for grabbing prey. Other limbs helped them walk, swim, or dig. In some species, the last pair of legs evolved into wide paddles that acted as underwater propulsion systems, letting them maneuver with surprising agility. Their mouthparts and feeding structures were equally varied. Some Euryterids were fierce predators, while others filtered small particles or scavenged dead matter. This flexibility allowed them to dominate different habitats and occupy many ecological roles. Euryterids weren't restricted to salt water. Fossils show they lived in freshwater rivers, coastal lagoons, tidal flats, and even brackish estuaries. Their ability to tolerate different salinities gave them access to ecosystems most marine predators couldn't reach. Some researchers believe certain species even ventured onto land for short periods, much like modern horseshoe crabs. Trackways from the Silurian and Devonian show arthropod footprints that match Euryterid limb patterns, suggesting they hauled themselves across beaches or mudflats, possibly during mating or to escape predators. Their sensory systems were advanced for the time. Large compound eyes provided wide fields of vision, and their bodies had fine hairs and sensory ridges that helped detect vibrations in the water. This made them effective ambush predators. They could wait silently, then strike with their pincers or rush forward with their swimming paddles. Smaller Euryterids hunted trilobites, worms, and early fish. Larger species likely tackled anything they could overpower, including other Euryterids. Despite their long success, Euryterids began to decline during the Devonian for several reasons. The rise of jawed fish, Placoderms first, then early sharks, introduced predators that were faster, better armoured, and more efficient hunters. These fish could crush Euryterid exoskeletons or outrun them in open water. At the same time, new marine ecosystems began forming around coral reefs, which favoured animals better suited to complex three-dimensional environments. Euryterids, with their flat bodies and limited climbing ability, were less effective in these new habitats. Climate change also played a major role. As continents shifted, coastlines changed shape, sea levels rose and fell, and freshwater systems became more variable. Euryterids relied heavily on shallow, calm environments, and disruptions to these ecosystems fragmented their populations. Many species became isolated and eventually disappeared. By the Carboniferous, only smaller freshwater Euryterids remained, and their once great diversity had shrunk dramatically. During the Permian, huge volcanic events caused the climate to become unstable, the oceans to become more acidic, and shallow water environments to disappear. Euryterids were already weak from conflict and natural stress, so they did not make it through the end Permian extinction. They disappeared completely, leaving only horseshoe crabs and spiders as distant cousins. These animals are in the same chelicerate group as them, but they don't have the same body plan. Ostracoderms, armoured, jawless fish. Ostracoderms were the first creatures that really moved toward becoming the animals we know today, like fish with backbones, sensory systems, and protection to protect them. They lived in the Ordovician, Silurian, and Devonian eras, a long time before sharks or other bony fish got mouths. These early fish were very different from current fish. There were no paired fins on their tails, and their mouths were only big enough to suck in soft food. They lived at the bottom and moved slowly. Still, for millions of years, they were the most common species in the oceans and rivers of the past. Their story is one of early ideas that didn't work out in the end, because jawed fish came along and changed everything. The most striking feature of Ostracoderms was their armour. Their heads, and sometimes their entire bodies, were encased in thick bone plates. These plates protected them from early predators, such as Euryterids, which hunted anything soft or unprotected. Armour also helped stabilise their bodies in water, since Ostracoderms lacked the internal skeleton structure that later fish developed. Their head shields often had elaborate patterns, ridges, and openings for sensory canals. some of the earliest examples of vertebrates experimenting with protective designs. Ostracoderms also had one of the first lateral line systems in the vertebrate world. This structure, still found in modern fish, allows animals to sense vibrations and water movement around them. For Ostracoderms, this sensory network was essential because they had poor eyesight and limited mobility. They relied on feeling their environment more than seeing it. Many species had eyes positioned on top of their heads, suggesting they spent much of their time partly buried in sediment, waiting for food particles to drift by. Their feeding method was simple, but effective for their time. Because they lacked jaws, Ostracoderms could not bite or chew. Instead, they fed by sucking in organic debris, plankton, or soft-bodied organisms from the seafloor. Some had small, tooth-like structures inside their mouths, but these weren't true teeth, just hard surfaces for scraping. This lifestyle worked well in calm, nutrient-rich environments. As long as the seafloor was quiet and predators were manageable, Ostracoderms had little competition. The insides of their bodies are one of the most unique parts of their biology. One of the first mammals to have a real backbone was an Ostracoderm, though it wasn't fully ossified like vertebrates do today. Along with this, they made early versions of gills, heart systems, and kidneys, which were important building blocks that later animals improved. These animals helped paleontologists figure out where the body plans of vertebrates came from by showing where certain systems first showed up and how they changed over time. Their evolutionary failure began when the first jawed fish, the Placoderms, and early sharks appeared in the Silurian and Devonian. Jaws were a major breakthrough. Instead of passively sucking food, jawed fish could bite, crush, tear, and grasp. This allowed them to become active hunters, pursuing prey rather than waiting for it. Placoderms such as Dunkleosteus could bite through bone. Early sharks could outswim almost anything. Compared to these agile predators, Ostracoderms were slow, heavily armoured and poorly equipped to compete. Jawed fish also developed paired fins, which gave them better balance and manoeuvrability. Ostracoderms lacked these completely. Their swimming was limited to wiggling their tails or moving their bodies in stiff motions that didn't produce much speed. As jawed fish spread across rivers and oceans, Ostracoderms were pushed into shrinking habitats where food was limited and competition was intense. Changes in the environment made the pressure worse. During the Devonian, also known as the Age of Fishes, climates changed and plant life on land grew quickly. This caused rivers and lakes to change dramatically. Sediments flowed into rivers, ecosystems became more complicated and predators varied. It wasn't enough for Ostracoderms to live a simple life like they used to. There were no more of them by the end of the Devonian. Acanthodians, spiny sharks. Acanthodians, which were sometimes called spiny sharks, even though they weren't really sharks, had a very strange mix of traits. They first showed up in the Silurian period and did very well through most of the Devonian. They populated lakes, rivers, and small seas with fish that looked sleek and dangerous, but were actually some of the earliest animals with jaws. Their name comes from the sharp spines that ran along each pair of their fins, which made them look like smooth-skinned sharks full of needles. For millions of years, Acanthodians tried out different body shapes and ways of eating. They were successful at first, but in the end, they all disappeared, leaving no living relatives. They came up with new ideas quickly, ruled for a long time, and then slowly went extinct as other fish changed faster than them. The thing that made Acanthodians stand out was the hard, pointed structures that stuck out in front of their fins. The cartilage in modern sharks is bendable, but these spines were stiff and often had layers of dentine covering them. They probably did double duty as defense and support, making the fins stiffer to make the animals more stable and scaring away predators. In many species, the spines had small lines or ridges on them, which suggests that Acanthodians were playing around with fin shape long before modern fish got it just right. Acanthodians had a strange blend of features from both bony fish and cartilaginous fish. Their skeletons were largely cartilaginous, like modern sharks, but they also had bony scales and small ossified elements in their skulls. These mosaic traits make them valuable to paleontologists because they help trace the early evolution of jaws and paired fins. Many Acanthodians had large eyes, suggesting good vision, and their streamlined bodies were built for speed in open water. But despite their fierce appearance, most Acanthodians were not top predators. Instead, many were plankton feeders, using specialized gill structures to strain small particles from the water. Some Acanthodians, such as Climatius, were agile swimmers that chased small prey. Others, like Acanthides, had toothless mouths and fed entirely on plankton. This variety shows how adaptable they were, occupying both predator and filter feeder niches in ancient ecosystems. Their gill structures were particularly advanced, with internal support bars that hint at the early stages of the bony fish gill skeleton. In this way, Acanthodians helped shape the basic anatomy used by most modern fish. A major breakthrough associated with Acanthodians was the refinement of paired fins. Although they didn't have full fin webs like modern fish, the pattern of their spines shows symmetrical right-left pairs that represent early steps toward the forelimb and hind limb structure of later vertebrates. Some researchers believe Acanthodians played a key role in the origin of paired appendages, an evolutionary leap that eventually enabled the rise of tetrapods on land. Despite their importance, Acanthodians faced rising competition during the Devonian. The oceans and freshwater systems became more dominated by advanced fish with stronger jaws, better teeth, and more efficient swimming techniques. Early bony fish developed rapid maneuverability and more precise fin control. Sharks evolved flexible cartilaginous skeletons perfect for speed. In this crowded environment, Acanthodians were increasingly pushed into smaller ecological spaces. Environmental shifts also added pressure. The Devonian period saw massive changes in river systems, sea levels, and oxygen availability as land plants spread across continents and altered global nutrient cycles. Acanthodians, many of which depended on plankton-rich waters, were vulnerable to these fluctuations. As plankton communities changed and new predators evolved, Acanthodian populations began to shrink. By the Carboniferous, only a few Acanthodian families remain. These survivors lived mostly in freshwater systems, but even there they faced competition from early ray-finned fish, which were more efficient filter feeders and swimmers. Over time, Acanthodians declined further, and by the early Permian, they were gone entirely. When they died out, there were no straight offspring left behind. There are no sharks or other bony fish to pass on their unique mix of traits, but they still had a big effect on the development of vertebrates. At a very important time when fish variety was growing quickly, they helped improve mouths, fins, sensory systems, and body forms. They show a time in fish development when a lot of different ideas were tried out quickly, but not all of them could survive long-term changes in the environment. Placoderms. Placoderms were the armoured giants of the Devonian seas. They were jawed fish covered in thick bony plates that had sharp jaw blades and were designed to rule a world where vertebrate predators were still in their early stages. They first showed up in the Silurian period, became very diverse during the Devonian period, and then went extinct almost overnight during the late Devonian extinction. Placodonts had a very short evolutionary history compared to groups like sharks or bony fish, but they left a huge mark on evolution. They were the first vertebrates to have real jaws, the first big predators to control sea environments, and some of the first animals to show signs of fertilization inside the body. Even so, for all their progress, they are the result of a huge evolutionary experiment that failed, leaving behind no straight descendants and no live equivalents. The most famous Placoderm, Dunkleosteus, was a giant among giants, up to 30 feet long, with a head covered in thick, interlocking armor plates. Instead of teeth, it had two massive jaw blades that formed a natural guillotine capable of slicing through bone. Studies of its biomechanics suggest that Dunkleosteus could generate bite forces rivaling modern crocodilians, with jaw speeds fast enough to snap shut in milliseconds. This combination of speed and crushing power made it one of the most dangerous animals of the Paleozoic. But Dunkleosteus was just one branch of a wildly diverse group. Placoderms came in many forms. Some, like the Arthrodires, were open-water hunters with jointed neck armour that let them lift their heads, an ability no earlier fish had. Others, like the Antiarchs, were bottom dwellers with box-shaped heads and armoured forelimbs. Some Placoderms lived in rivers, some in shallow seas, and others in reefs. Their body plans range from flattened skimmer-like forms to bulky, deep-bodied swimmers. This diversity suggests that Placoderms were experimenting rapidly with different feeding strategies, swimming styles, and habitats at a time when vertebrate evolution was still highly flexible. What makes Placoderms so important is their jaw innovation. Jaws evolved from modifications of the first gill arches, and Placoderms were the earliest vertebrates to perfect this transformation. Their jaws were supported by strong muscles and reinforced by armour, giving them powerful biting capabilities. Without Placoderm jaws, later vertebrates, including sharks, bony fish, reptiles, mammals, and humans, would not have the complex mouth structures they rely on today. Placoderms introduced a revolution in how animals could feed, hunt, and interact with their environment. Placoderms also show some of the earliest evidence for internal fertilization. Fossils of species like Matopechis attenboroughii contain embryos preserved with umbilical attachments, proving that live birth existed long before sharks or mammals adopted it. This means Placoderms weren't just experimenting with armour and jaws, they were rewriting reproductive strategies in ways that would later evolve in completely different groups. Their embryos and reproductive structures reveal a complexity that contrasts sharply with their bulky, armoured exteriors. But despite their innovations, Placoderms faced rising challenges during the late Devonian. New groups of fish, especially early sharks and ray-finned fish, began evolving faster and more flexible bodies. These newcomers lacked heavy armour, giving them superior speed and manoeuvrability. As ecosystems became more complex, mobility became more important than defence. Placoderms were powerful but inefficient. Their heavy armour restricted rapid turns and long-distance swimming. Meanwhile, sharks refined their cartilage skeletons into incredibly flexible frameworks, and ray-finned fish evolved lightweight scales and versatile fin designs. Environmental upheaval accelerated Placoderm decline. The late Devonian saw ocean anoxia events, periods where huge stretches of ocean lost oxygen. Armoured fish, depending on stable shallow habitats, were hit hardest. Reef systems collapsed, coastal waters fluctuated, and nutrient cycles changed as land plants altered global environments. Placoderms, which had once filled every available niche, suddenly found themselves squeezed by both environmental stress and faster, more adaptable competitors. During the late Devonian extinction, there were no more Placoderms. Through the Carboniferous, not a single branch made it. Sharks and bony fish, which were their main rivals, lived, diversified, and eventually took over every aquatic environment on Earth. Even though platycerms evolved a lot, they ended up becoming a dead branch. Antiarchs. Antiarchs were some of the weirdest animals that lived in the Devonian. They were armoured fish that walked on the bottom and looked like a mix between a turtle, a tank, and a mechanical toy. Antiarchs were small, heavily armoured bottom feeders that were part of the larger Placoderm group. Unlike the scary hunters like Dunkleosteus, they had strange structures that looked like limbs and worked more like fingers than fins. They lived all over the world in rivers, lakes, and small seas. They did very well for tens of millions of years until the late Devonian extinction, when they were all gone. Their strange shape is the result of a risky evolutionary experiment that looked at how early vertebrates explored life on the seabed. Unfortunately, the experiment finished in a dead branch. The best-known Antiarch, Bothriolopus, was about the size of a loaf of bread, though some species were larger. Its body was divided into two main regions, a heavily armoured front section and a soft, flexible tail. The armoured portion was made of thick bone plates arranged like interlocking shell, giving Antiarchs their signature boxy appearance. These plates protected their vital organs from predators and also served as structural support in environments with strong currents or shifting sediments. But the most iconic feature of Antiarchs was their pectoral appendages, elongated, jointed structures extending from the sides of their armoured box. These weren't fins in the modern sense. Instead, they resembled stiff, bony arms that could bend at the elbow and move in coordinated motions along the seafloor. Fossils show wear marks that suggest Antiarchs used these appendages to prop themselves up, steer through muddy bottoms, or even walk across riverbeds. This unusual form of locomotion was a major evolutionary experiment in limb development long before tetrapods evolved legs. Their feeding strategies were equally unusual. Antiarchs had small mouths located on the underside of their armoured heads, positioned perfectly for grazing along the seafloor. They likely sucked up detritus, algae, or small organisms hidden in sediment. Their jaws were simple compared to other placoderms, but efficient enough for a bottom-feeding lifestyle. Their eyes sat near the top of their heads, giving them a wide field of view to watch for predators while they fed. This eye placement resembles modern bottom dwellers like sturgeons and rays. Antiarchs were found all over the world. Fossils have been found in Australia, Europe, Asia, and North America. This suggests that they were very flexible and could live in a wide range of places, from freshwater rivers to coastal bays. During the Devonian, predators like Euryterids and early sharks were becoming more popular. Their heavy armour may have kept them safe. However, their armour made them slow and hard to move around. These traits helped in calm, safe environments, but hurt them when ecosystems changed. Recent studies using CT scans and 3D reconstruction have revealed that the internal anatomy of Antiarchs was more complex than previously thought. Their pectoral appendages had intricate joint surfaces that allowed controlled movement, not just simple flexing. This level of limb functionality shows that vertebrates were experimenting with limb-like structures long before limbs were used for walking on land. Although these structures never led directly to tetrapod leg, they demonstrate how evolution explored multiple approaches to movement. So why did Antiarchs fail? Part of the answer is competition. During the Devonian, ray-finned fish and lobe-finned fish diversified rapidly. These fish had more flexible fins, better swimming abilities, and more efficient feeding structures. As rivers and lakes became more crowded with fast, agile competitors, Antiarchs were pushed out of many niches. Their armour, once a strength, became a burden when speed and flexibility became more important for survival. Environmental changes also played a major role. The late Devonian saw drastic shifts in water temperature, oxygen levels, and sediment flow as land plants spread and altered global nutrient cycles. Many shallow water habitats became unstable or disappeared entirely. Antiarchs, which depended on these calm environments, were hit hard. When the late Devonian extinction events triggered widespread anoxia in aquatic systems, Antiarchs were among the groups that couldn't recover. Diadectids, stem amniota herbivores. Diadectids were some of the first big plant-eating vertebrates to walk on land. They were big, strong animals that lived in the late Carboniferous and early Permian, a long time before reptiles and mammals took over. They were neither true snakes nor true frogs. Instead, they were in the middle of development as stem amniotes, which means they were related to the first mammals that evolved fully shelled eggs. For millions of years, Diadectids did well as heavy herbivores, creating the first environments on land. But even though they were important, their whole family tree disappeared before the Mesozoic, leaving only bones and questions about why such powerful animals didn't have any children. The most famous member, Diadectes, was built like a bulldog crossed with a tortoise. It had a massive skull with blunt, chisel-shaped teeth for grinding plants, some of the earliest true herbivore teeth in vertebrate history. Before Diadectids, most land vertebrates were small insect hunters or carnivores. Plants were abundant, but few animals were adapted to digest them. Diadectids changed that. Their broad ribs and barrel-shaped bodies housed long digestive tracts capable of breaking down tough plant fibers, giving them access to an enormous and untapped food supply. This single shift marked a turning point on land. Vertebrates were no longer limited to a meat-first diet. Their skeletons reveal an animal well adapted for a slow but powerful lifestyle. Diadectids had thick limb bones, wide feet, and strong shoulder and hip joints that supported their heavy frames. Unlike earlier amphibians, They held their limbs more directly beneath their bodies, allowing for more stable movement on land. They weren't fast, but speed wasn't necessary. Their size, strength, and bulk gave them protection from predators like early synapsids and large amphibians. Some species grew more than 2 meters long, making them some of the biggest land animals of their time. The place of Diadectids on the genetic tree is one of the most interesting things about them. They lived very close to the first real amniotes, which were animals that could lay eggs with shells that protected them so babies could grow on land instead of in water. However, Diadectids were not really amniotes. It has been discussed for decades about how to best classify them. Some paleontologists say they were just outside the amniote crown group and developed many of the same traits on their own. Some people think they might have been very early, basic amniotes that lost some of their traits. No matter which explanation is right, they show a very important time in the development of vertebrates, the change from reproducing in water to living on land. Their jaws were another evolutionary milestone. Diadectids had robust jaw muscles and complex tooth structures with wear surfaces. Clear evidence of chewing. True chewing is rare in early vertebrates. Most reptiles and amphibians swallow food whole or with minimal processing. Diadectids were among the first creatures to actually grind vegetation to extract nutrients. This chewing behavior foreshadowed later herbivores like dinosaurs and mammals, which would turn chewing into a major evolutionary advantage. But their success couldn't last forever. As the Permian progressed, the world shifted dramatically. Global climates became drier, forests thinned, and new plant groups emerged. At the same time, synapsids, the ancestors of mammals, were diversifying. Early synapsids, like Edaphosaurus and Cassia, developed more advanced herbivore adaptations, including lighter skeletons, more efficient metabolisms, and better temperature regulation. These new herbivores were simply more adaptable than Diadectids, which relied on heavy bodies and slow movement. Predation pressures also increased. Large synapsioid predators, such as Dimetrodon, evolved serrated teeth, faster gaits, and better sensory systems. Diadectids, with their slow speed and bulky frames, may have struggled to escape or defend against these increasingly specialized hunters. Their armorless bodies offered little against slicing jaws. Environmental stress accelerated their decline. As rivers dried and swampy forests gave way to open, seasonal landscapes, Diadectids lost the stable habitats they depended on. Their physiology was suited to moist environments with abundant vegetation, not the patchy, fluctuating ecosystems that dominated the late Permian. They were no longer around by the early Permian. Their entire family died out, leaving no living branches behind. It was a clever genetic experiment that worked for millions of years, but it couldn't keep up with faster, more efficient herbivores and climates that changed quickly. Lepospondyls. Some of the odd early tetrapods were lepospondyls. They were small, varied, and often shaped in ways that make them look more like mythical creatures than real ancient animals. The Carboniferous and Permian times were when they lived. They tried out a lot of different body types before lizards and mammals took over the land. Diplocaulus, the most famous lepospondyl, was a totally different animal. It was an amphibian with a boomerang-shaped head and a brain that is so wide and triangular that it has stumped paleontologists for more than 100 years. Lepospondyls are an example of a lot of trying new things in the early stages of tetrapod development. These attempts failed when more advanced animals beat them. Diplocaulus, the best-known member of this group, had a skull shaped like a giant V, with two long horn-like projections extending outward. This bizarre shape wasn't ornamental. Recent studies suggest the skull acted as a hydrofoil, helping the animal glide through the water with greater lift and stability. As it swam, water flowed along the skull's surfaces, generating upward force and keeping the animal level in the water column. This made Diplocaulus surprisingly agile, able to dart away from predators or ambush prey from below. It was a clever adaptation, one not seen in any modern amphibian. Other lepospondyls explored completely different strategies. Some, like Ostracoderm, were long and limbless, resembling early snakes or oversized worms. They moved by undulating through tight spaces, which suggests they hunted small prey in burrows or dense vegetation. Others, such as Nectridea, had flattened bodies and paddle-shaped tails for swimming. Still others were stubby-bodied and armoured, built for life on muddy stream banks. This diversity shows that Lepospondyls were not a single type of animal, but a whole cluster of evolutionary experiments branching off from early tetrapod. Their skeletal structure sets them apart. Lepospondyl vertebrae were formed from a single piece of bone called a lepospondyle centrum, rather than the multiple fused elements typical of other early tetrapods. This gave their bodies extra stiffness and simplicity, which may have been advantageous in small-bodied animals. Their skulls were also more lightly built than those of early amphibians, suggesting they relied more on flexibility than brute strength. Despite these innovations, Lepospondyls never achieved large sizes. Most were under a foot long, and even the largest rarely exceeded a metre. Their small size made them vulnerable to predators, especially as early reptiles and amphibians grew larger and more specialised. The rapid evolution of terrestrial predators, such as large temnospondyle amphibians, may have squeezed Lepospondyls into narrower ecological niches. Lepospondyls thrived in the warm, swampy environments of the Carboniferous, but as the Permian progressed, global climate shifted toward drier, more seasonal conditions. Wetlands shrank, river systems changed, and freshwater habitats became less stable. Lepospondyls, many of which depended on aquatic or semi-aquatic lifestyles, struggled in these fluctuating landscapes. Their delicate bodies and specialised swimming adaptations became disadvantages as environments became harsher. Another factor was competition from the early amniotes, reptiles and their close relatives. Amniotes evolved waterproof skin, more efficient lungs, and eggs that could be laid on land. These traits allowed them to spread into dry habitats inaccessible to Lepospondyls. As amniotes diversified into insect eaters, herbivores, and predators, Lepospondyls lost ground. Their simpler vertebrae and soft bodies couldn't keep up with the stronger skeletons and more advanced physiology of their competitors. What's unique about Lepospondyls today is how different and creative they were during a very important time in the development of tetrapods. Long before reptiles and animals improved the tetrapod pattern, they tried out different body shapes and ways of moving. Their mistakes teach us just as much about evolution as their achievements. They show that the past of early tetrapods wasn't a straight line leading to modern amphibians and reptiles, but a complicated web of forms that were tried and failed. Giant lycopsid trees, Lepiododendron forests. In the late Carboniferous, huge lycopsid trees like the famous Lepidodendron used to grow together to make woods that covered a lot of land. The trees didn't look at all like woods do now. They had diamond-shaped scales instead of bark. They didn't grow branches. Instead, they made tall poles that split near the top. Instead of making wood, they used soft, flexible tissues to build their stems, which helped them grow very quickly in wet, swampy places. Lycopsid forests were the most important plant environments on Earth for millions of years. They changed the temperature and made a lot of the coal we burn today. Even though they did well, these strange trees were just part of an experiment in evolution that failed when the planet dried out. A mature lepidodendron could reach 30 to 40 metres tall, similar to a modern redwood, but it grew in a completely different way. Modern trees thicken over time through secondary growth, adding rings of wood each year. Lycopsids did not. Their trunks were hollow in the centre and filled with soft tissues instead of wood. This meant they grew fast but were structurally fragile. They relied on the constant support of swampy soils, where their roots could spread wide. Their roots, called stigmarian systems, were actually modified underground stems covered in rootlets. These structures formed huge networks that stabilized the trees in muddy ground. Lycopsid leaves were long, narrow structures that sprouted directly from the trunk when young. As the tree matured, older leaves dropped off, leaving behind the distinctive diamond scars we see in fossils. Near the top, lycopsids formed crown-like clusters of leaves. From these crowns grew massive spore cones, some bigger than a human arm. Lycopsids reproduced using spores instead of seeds, meaning they were tied to humid, wet environments where spores could disperse easily. How quickly they grew up is one of the most interesting things about lycopsid nature. Based on fossils, these huge trees only lived 10 to 15 years, which is incredibly small for their size. They grew very quickly and reached amazing heights. They only reproduced once, and then they died soon after, sending out germs. Because they lived for such a short time, they were perfect for the huge, wet coal swamps of the Carboniferous, where conditions were unstable because of frequent floods and sediment building. Because they grew quickly, lycopsids could quickly take over new places and beat out plants that grew more slowly. But this specialization was also their downfall. As the Carboniferous came to an end, global climates began shifting. The supercontinent Pangaea was forming, creating larger landmasses with more interior regions far from moisture. Rainfall patterns changed, seasonal climates appeared, wetlands shrank, coal swamps dried out, and lycopsids, tied entirely to saturated soils and spore-based reproduction, had no way to adapt. Their entire ecosystem began collapsing. At the same time, seed plants, early gymnosperms, were rising. Seed plants could survive in dry environments. Their seeds protected embryos and could remain dormant during harsh conditions. They developed stronger wood, deeper roots, and more flexible growth patterns. These advantages allowed gymnosperms to move into upland areas, hillsides, and drier terrains that lycopsids could not tolerate. As forests diversified, gymnosperms pushed lycopsids into the shrinking wetlands that remained. Once swamp environments became patchy and unpredictable, the lycopsid life cycle broke down. Without stable wetlands, spores couldn't spread successfully. Without wet soil, their shallow stigmarian roots couldn't support the trees. Their towering, fast growth strategy, which once made them unbeatable, became a fatal flaw. By the early Permian, giant lycopsids had disappeared from Earth. Even though they went extinct, their legacy lives on in a literal, physical way. The coal seams beneath our feet. As lycopsid forests collapsed and were buried under sediments, their soft tissues compressed over millions of years into coal. Much of the coal used during the Industrial Revolution, and still used today, comes from the ancient remains of these lost forests. Gorgonopsians, saber-toothed proto-mammals. It was more than 250 million years ago, long before dinosaurs, and the Gorgonopsians were the top hunters of that time. These animals were not reptiles or mammals. Instead, they were early synapsids, which are related to the branch that would eventually become mammals, but are very different from them. With long teeth that looked like sabers, eyes that looked forward, and bodies that were made for running across rough floodplains, Gorgonopsians were the best hunters on land in the last few million years before the biggest extinction event in history wiped them out. Their rise and fall shows one of the most dramatic tests in evolution that took place long before mammals evolved, making warm-blooded hunters. The general image of a Gorgonopsian is Inostrancevia, a 2-meter predator with enormous slicing teeth and a skull built for delivering deep, fatal bites. But the group as a whole was diverse. Some were the size of foxes, others were nearly tiger-sized. All shared key traits: elongated canine teeth, lightly built limbs for fast movement, and skulls shaped to deliver powerful bites. Unlike earlier amphibian-like predators, Gorgonopsians had fully upright postures, allowing them to run rather than crawl. Their limbs tucked beneath their bodies, suggesting higher activity levels and possibly even warm-blooded metabolisms. Their skulls were engineering marvels for their time. Gorgonopsians developed some of the earliest true saber teeth, long and laterally flattened canines used for precision killing. Their jaw joints allowed wide gapes. Some species could open their mouths nearly 90 degrees, far wider than modern big cats. This gape was necessary to use their sabers effectively. Bite marks in fossil bones suggest they targeted soft tissues and major blood vessels, much like later saber-toothed mammals would. This convergence, saber teeth evolving separately in unrelated groups, shows that evolution often revisits successful designs in different eras. Gorgonopsians also demonstrate early steps toward mammal-like sensory and muscular systems. Their skulls show enlarged attachment areas for jaw muscles, allowing fast bite closure. Their eyes faced more forward than those of many reptiles, improving depth perception for hunting. Some species may have had whisker-like sensory systems, inferred from facial bone patterns similar to those in early mammals. Their rib structures also suggest a more flexible breathing system, possibly allowing sustained activity and greater endurance than their contemporaries. Their environment demanded this level of adaptation. During the late Permian, the supercontinent Pangaea created extreme climates, scorching summers, long droughts, and unpredictable rains. Floodplains alternated between lush growth and barren dryness. Herbivores such as Dicynodonts and Paraeosaurs roamed these landscapes, forming the prey base that Gorgonopsians depended on. Many prey species had armor-like bony projections, or tough hides, so Gorgonopsians relied on precision strikes rather than brute force. Their sabers delivered those strikes efficiently, but their specialization came with risks. Saber teeth were powerful tools, but they were also fragile. If a canine broke, a Gorgonopsian might struggle to kill prey effectively. Their long legs and lightly built bodies made them excellent hunters, but not as well suited for grappling with heavily armored herbivores. Their dominance lasted only as long as the ecosystem remained stable enough to support large, active predators. The end Permian mass extinction, which was the worst extinction event in Earth's history, broke up that security. Huge volcanic events in Siberia caused acid rain, global warming, seas without oxygen, and the breakup of food webs. Ecosystems broke up on land. First the plants died, then the animals, and finally the predators that needed them stopped existing. Gorgonopsians, which were at the top of the food chain, were some of the first big land animals, never to be seen again. Their very specific way of life as predators didn't give them much freedom when food became scarce. No Gorgonopsian survived into the Triassic. Their disappearance opened ecological space for the next major lineage of synapsid predators, therapsids like therocephalians, and later cynodonts, which would evolve traits even closer to mammals. But none of these later groups ever recreated the exact form or function of Gorgonopsians. Their sabers, their posture, and their predatory style were unique to the late Permian, a brilliant but temporary evolutionary design. The animals that lived on Earth before dinosaurs show that evolution doesn't go in a straight line. Every time the planet moves, evolution makes new discoveries, throws out old ones, and changes the rules. From huge coal swamp trees to saber-toothed early animals, each branch that died out made room for the next one to grow. Please like, subscribe, and join us for the next video if you like this trip into the lost experiments of deep time. The world of the prehistoric times is full of stories more bizarre than the ones that didn't make it.